A few seconds ago, without consulting you, your brain made a choice. Your body settled into whatever it was resting against. Your breathing slowed without you noticing. At some point, the signal to keep listening was sent and you kept listening. None of these felt like decisions. They felt like stillness, like the body knowing what to do without being asked. But here is what the research has been carefully demonstrating for decades. By the time you became aware of making a choice, the physical process of making it had already begun. The neural signature of the decision appears in your brain before you report any experience of deciding, in some cases by several hundred milliseconds, in others by several seconds. This is not a philosophical argument. It is a measurable physical fact about what happens inside your skull between the moment a decision begins and the moment you feel yourself making it. Think about what you use decisions for. You use them to navigate the world, to choose between options, to commit to actions that ripple outward. You experience the moment of deciding as the cause of what follows. The intention forms, and then you act on it. Thought first, then deed. In that order, or so it feels. What the brain shows is something different. The preparation for the action is already underway when the awareness of intending it arrives. You feel like the author of your choices. What the record in your neuron suggests is something closer to a reader who arrives at the end of a sentence the brain has already written. You build your sense of yourself around your choices. The idea that you are the author of what you do is not peripheral to your identity. It is close to the center of it. When you hold yourself responsible for something you did, or take pride in a good choice, you are treating the felt experience of deciding as real, as the thing that counts, and the neuroscience does not straightforwardly agree. But neither does it say you're simply a passenger. The relationship between what the brain does before you are aware of it and what you experience as conscious choice is more subtle than a simple verdict either way. The brain that decides in the dark and the mind that experiences deciding are the same organ, working at different speeds, at different depths. The brain is doing this constantly, at the level of which eye to let your gaze settle on, at the level of whether to swallow right now or wait, at the level of whether a thought forming in the background is worth attending to. Most of these never reach you as choices. They are resolved quietly, and the outcome is simply the next moment of your experience, already assembled and delivered. The ones that reach you as choices, the ones where you feel the deliberation and the weighing and the moment of commitment, are a small and arguably unrepresentative sample of what the brain is deciding on your behalf at any given second. What is a decision physically? What does it look like when the brain builds one from the competing noise of options and past experience and the felt pull of the body and arrives at a single committed action? What fires, in what order, and who is actually in charge? If you enjoy exploring questions like these about the brain and the mind, a like would be highly appreciated. And if something about this question raises a thought worth sharing, or something you have noticed in your own experience of making choices, the comments are open for exactly that. With that said, let's get back to the topic. Think about what you were doing in the moment before you last moved. Before you reached for something, stood up, shifted in your chair. You probably experienced it as a single event, a brief sense of intention followed by the movement itself, the intention and the action, feeling like two parts of the same thing, arriving together. The brain's record looks nothing like this. Before any single voluntary action, several brain areas are already active. Your supplementary motor area is processing one version of the movement. Your premotor cortex is running another. Your parietal cortex is calculating the reach, the distance, the angle. These are not sequential steps in a conscious planning process. They happen simultaneously, in parallel, in regions distributed across the cortex. Each one is generating a proposal for what you might do next. Most of those proposals are never acted on, but they are all running, all the time. There is an idea that most people carry, usually without examining it, about how a decision works. In this picture, the mind perceives a situation and weighs the options until a preference emerges. Then it hands that preference to the body as an instruction. Thought first, action second. The decision as the cause and the movement as the effect. This picture is intuitive, and it fits the way choices feel. It is also significantly incomplete. A neuroscientist named Paul Chisek, working at the University of Montreal in the early years of this century, named what he saw in the data differently. He called it affordance competition. The idea was this: the brain does not wait until it has decided what it wants to do before beginning to prepare for action. Instead, it prepares several possible actions at once, while it is still gathering the information it needs to choose between them. Preparation and selection run in parallel, not in sequence. The body is already partially in motion for several different futures at the same moment that the brain is still deciding which future to commit to. Chisek arrived at this framework partly by studying how both monkeys and humans respond when a decision changes at the last moment, when a target they were about to reach for suddenly moves. What he found was that the motor system can update its preparation almost instantly, too quickly for a sequential model to explain. The motor proposals were already in motion. Redirecting them required only a small shift, not a full restart, which meant the preparing had come before the choosing, not after. Imagine an orchestra in the moments before a performance begins. Every musician is already in position, instrument raised, ready to play. The violinists have their bows on the strings. The players at the woodwinds have their instruments at their lips. The percussionists are poised above their drums. No sound has been produced yet, but every section is already primed. All of that readiness is in place simultaneously. The performance does not start with one section preparing first while the others wait. All of them are prepared at the same moment, in the same breath. The question is only which section the conductor signals to begin. Your motor system works like this all the time. At every moment, many possible next actions are partially prepared in different brain areas. The system is not idle, waiting to be told what to do. It is always already generating proposals in parallel, each one representing a different route the body might take. At any instant, you are preparing to reach, to stand, to speak. All of those proposals are live simultaneously. The one that eventually happens is not the only one that was ready. A question follows from this. If multiple competing proposals are always in motion, how does one of them get selected? What is doing the selecting, and how does it know when to act? The brain has a dedicated structure for exactly this job, and the way it carries out that selection is nearly the inverse of what most people would expect. It does not weigh the proposals and pass a winner down the line. It suppresses everything by default and then releases exactly one thing. The moment of deciding is not a moment of adding something. It is a moment of removing what was in the way. The name for this structure is the basal ganglia, and it sits at the base of your brain, a dense cluster of interconnected regions buried beneath the cortex. It receives input from nearly every part of the brain that deals with planning and movement. And what it does with that input is not what most people would guess. The common assumption is that the brain selection process works like a voting machine. The proposal with the most support wins, a positive signal is sent and the action is triggered. Something like a recommendation passed down until it reaches the muscles. The basal ganglia works like the opposite of this. Its default state is suppression. Deep inside the circuit, two structures fire continuously: the globus pallidus and the substantia nigra. They send inhibitory signals to the thalamus. The thalamus, which sits between the deeper brain and the cortex, is being held quiet. Held quiet means that the cortical motor proposals, the competing signals your premotor and supplementary motor areas are generating at this very moment, cannot easily break through into action. The gate is closed. Not because nothing is happening, but because the thing that would let anything through is being kept down. Imagine a railway station where every platform operates with a locked gate. The trains are ready, signals green, engines running. But every gate is held shut by default by a central mechanism that is always on. The gates do not open because a new instruction tells them to open. They open because the mechanism holding them shut is briefly disabled. A specific lock is released. One gate swings open. The train leaves. All the other gates remain closed. That is disinhibition, and it is the mechanism the brain uses to select action. The striatum is the main input hub of the basal ganglia. When it receives enough excitatory signal from the cortex for a particular action, it activates. This activation travels down what is called the direct pathway, and it suppresses the globus pallidus. The globus pallidus, now suppressed, can no longer hold the thalamus down. The thalamus becomes more active, and the motor signal for that one action breaks through. But while that one gate opens, a parallel route through the indirect pathway is simultaneously strengthening suppression on the competing actions. The winning signal does not just get through. The losing signals get pushed down harder at the same moment. The selection is not just a release of one proposal, it is a focused removal of what was stopping one thing, combined with an increase of what is stopping everything else. One gate opens, all the others close tighter. A neurologist named Malin DeLong spent decades mapping this circuit, recording from single neurons inside the basal ganglia of monkeys with Parkinson's-like symptoms. What he found was that the subthalamic nucleus, part of the indirect pathway, was firing much too strongly. The result was excessive suppression across the board, making it nearly impossible for any signal to break through. The tremor and rigidity of Parkinson's disease are, in part, the body's experience of a selection system that has lost its ability to release one thing cleanly. Nothing gets through, or the wrong things do, too slowly, in the wrong order. DeLong's work eventually led to a treatment, deep brain stimulation, in which an electrode is placed into the subthalamic nucleus, and a small continuous electrical signal is used to quiet its overactivity. It does not add something. It removes the excess of what was blocking things. Even the treatment works by disinhibition. The selection is happening right now, below anything you are consciously doing. The basal ganglia is receiving the competing proposals from the cortex, running the direct and indirect pathways in parallel, and deciding which one should clear the gate. But how does it know which one to let through? How does one proposal become the winner? The answer depends on something the basal ganglia cannot compute on its own. It needs a signal that tells it how much to weight each option, how much the expected outcome of one action outweighs the others, and that signal does not come from reasoning. It comes from a chemical that most people know by entirely the wrong description. Dopamine has a reputation, and the reputation is wrong. Popular descriptions of this chemical usually land in the same territory. It is the brain's pleasure signal, the thing that lights up when something feels good, the molecule that drives enjoyment and reward. It is none of these things, or rather, it is something considerably more precise and considerably stranger. What dopamine actually encodes is not pleasure. It encodes expectation. To understand what that distinction costs, consider the experiment that established it. In the late 1980s, a neuroscientist named Wolfram Schultz was recording from single neurons in the brains of monkeys at the University of Freiburg. He was trying to understand how the brain's movement system worked, and the neurons he was tracking were in a region rich with dopamine-producing cells. He expected the cells to respond to the movements the monkeys were making. Instead, they were responding to something else entirely. When a monkey received an unexpected squirt of juice, the dopamine neurons fired. That, Schultz noted, made a kind of sense. Something good happened. The chemical associated with reward spiked. Straightforward enough. But then the experiment changed. Schultz trained the monkeys to associate a light with the delivery of juice. After enough repetitions, the monkeys knew that when the light appeared, juice would follow, and when that association was learned, the dopamine neurons stopped responding to the juice. They started responding to the light. The reward was the same. The dopamine signal had moved entirely to the prediction. Schultz sat with that finding for some time before its full meaning became clear. The dopamine system was not reporting on pleasure. It was not saying this is good. It was saying this is what I expected would lead to something good. The juice itself, once it was predictable, stopped being interesting to these cells. What was interesting was the signal that preceded it, the signal that promised it was coming. Dopamine was a signal about the future, not the present. Think of a forecaster who issues probability reports. Not descriptions of how it will feel to be caught in the rain, but an estimate of likelihood. The report says, based on conditions, something good is expected here. When conditions turn out to match the forecast, not much happens. The forecast was already priced in. But when conditions diverge from the forecast, when something better arrives than expected or something worse, that divergence is the signal that matters. Dopamine fires in exactly this pattern. It spikes when outcomes are better than predicted. It drops below its baseline rate when expected outcomes fail to arrive. When the prediction is exact, the response is flat. What this means for the basal ganglia is significant. The striatum, which receives the competing action proposals from the cortex, is also richly connected to dopamine-producing neurons in the midbrain. Dopamine activity modulates how strongly each competing proposal is weighted. An action that has been associated with better-than-expected outcomes in the past will be weighted more heavily. An action associated with disappointment will be weighted less. The gate that opens for a particular proposal is not random. It is biased by the history of what following that kind of proposal has previously led to. This is how the basal ganglia knows which proposal to release. The competing signals arrive. They carry implicit value weights, shaped by past experience and modulated by dopamine. The selection is not arbitrary. It is the brain running a fast, largely unconscious calculation about which available action is most likely to lead somewhere good, but that calculation has to come from somewhere. The value weights that dopamine applies are not built in, they are learned. And to understand how they are learned requires understanding what happens to the dopamine signal when reality and prediction disagree. Wolfram Schultz had discovered something about what dopamine signals, but there was a second layer beneath it. When the monkeys received juice exactly as predicted by the cue, the dopamine neurons were quiet. The prediction was met. Nothing unusual to report. But when the juice arrived at a moment the monkeys had no reason to expect it, the dopamine neurons fired sharply. And when the juice was expected but did not arrive, something equally striking happened. At the exact moment the juice should have come, the dopamine cells dropped below their baseline firing rate. They went quiet in a way that was itself a signal. The absence of the expected thing produced a response as distinctive as the presence of an unexpected good thing. What Schultz had recorded was not a pleasure signal. It was a prediction error signal, a positive prediction error, outcomes better than expected. Dopamine fires above baseline. A negative prediction error, outcomes worse than expected. Dopamine falls below baseline. Prediction exactly met. Nothing changes. The signal is not about what happened. It is about the gap between what happened and what was expected. This distinction matters because of what the dopamine signal does next. The signal travels from dopamine-producing neurons in the midbrain, in a region called the ventral tegmental area, to the striatum. The striatum is where the value weights on competing action proposals are stored. When dopamine arrives at a synapse associated with an action that just produced a better-than-expected outcome, that synapse gets slightly stronger. When dopamine drops because an expected outcome failed to arrive, synapses associated with that action get slightly weaker. Over many trials, the system converges on a set of value weights that reflect the actual probabilities of good outcomes attached to different actions. The brain is not computing reward, it is computing surprise about reward, and it is using that surprise to update its predictions for next time. In the mid-90s, 3 researchers noticed something striking about the mathematical structure of what Schultz had found. Their names were Peter Dayan, Red Montague, and Terry Sinowski. The pattern of dopamine activity matched a mathematical framework from computer science called temporal difference learning. Dopamine fired to cues, dropped when expected rewards were omitted, and was silent when predictions landed exactly. This was a method by which algorithms could learn to maximize future reward by updating predictions based on the difference between expected and actual outcomes. The equations were the same. The brain was doing the thing, and the computer science field had independently derived the math for it. The dopamine system in every mammal was running something functionally equivalent to a form of machine learning that engineers would not formalize until the 1980s. The brain had been doing it for millions of years. What this means is that the value weights feeding into your basal ganglia, the weights that determine which competing action proposals the gate will open for, are not fixed. They were built over the course of your life by an ongoing process of predicting outcomes, comparing those predictions to what actually happened, and adjusting the weights accordingly. Every choice you have made, and the outcome that followed, has contributed a small update to the probability map your brain now uses to guide the next choice. This is why familiar situations produce faster decisions. The value weights are well calibrated. The competing proposals are not equally likely. One of them carries a history of working, and that history has been written into the weights of the synapses that run the competition. But even a perfectly calibrated value system still needs to make the final call. The dopamine weights tell the basal ganglia which proposals to favour. They do not tell it when to commit. For that, the brain uses a different kind of computation entirely, one that trades off speed against accuracy, in a way you feel every time you have to decide quickly. There is a moment in any difficult decision when you feel yourself getting close to committing. Something tips. The sense of the options being open closes, and one of them becomes the one you're going to take. You may not be able to say exactly when it happened, but the tipping was real, and the brain has a physical mechanism for it. The mechanism is called accumulate to threshold, and it was found not in a human brain during a major life choice, but in the brain of a monkey watching moving dots. In the 1990s, a neuroscientist named Michael Shadlin designed one of the most revealing experiments in the history of decision neuroscience. He was working at the University of Washington at the time before later moving to Columbia. He showed monkeys a screen filled with randomly moving dots. Some of those dots moved coherently in one direction. Others moved at random. The monkey's task was to decide which direction the coherent dots were heading, and indicate that choice with an eye movement. Sometimes the signal was strong, most dots pointing clearly in one direction. Sometimes it was barely detectable, only a few dots moving coherently, the rest noise. While the monkeys made these decisions, Shadlin and his collaborator William Newsom were recording from neurons in a region of the parietal cortex called the lateral intraparietal area. What they found in those neurons was not what anyone had expected to be so visible in a single cell's activity. The neurons were accumulating. When the dots appeared, the relevant LIP neurons began increasing their firing rate. not all at once and not in a step-like jump. Gradually, like a slow climb. The rate of climb depended on how strong the motion signal was. When the coherent dots were numerous and clear, the climb was steep. When the signal was weak and noisy, the climb was shallow and variable. But in both cases, the climbing stopped the same way. When the firing rate crossed a threshold, the monkey made its choice. The threshold was fixed. The rate of approach to it varied. The decision formed in the climbing. Think of a bucket being filled by a stream of evidence. Each glimpse of coherent motion adds a small amount. Each glimpse of noise adds a small amount in the opposite direction, or nothing. The stream continues until the bucket fills. The decision does not happen when a particular drop arrives. The decision happens when the bucket tips, and whether that takes 100 milliseconds or 3 seconds depends on how fast the stream is running. This model, the drift diffusion model in its mathematical form, captures one of the most important properties of real decisions, the trade-off between speed and accuracy. Lower the threshold at which the bucket tips, and decisions come faster but with more errors because less evidence has accumulated. Raise the threshold, and decisions take longer but tend to be more reliable. You have felt this trade-off. Under time pressure, you decide with less information and sometimes get it wrong. Given time to deliberate, you are more likely to be right. The brain is not running an arbitrary process. It is running the mathematically optimal version of this trade-off for a given cost structure. The most striking thing Shadlin and Newsom's work revealed was that this process is not specific to moving dots. The same ramping to threshold pattern shows up in neurons involved in value-based decisions, in decisions about social actions, in decisions about risk. The accumulation process appears to be a general feature of how the brain computes decisions across many different domains. It is the common mechanism underneath the diverse surface of choices. And here is where the physical record of deciding becomes genuinely strange. Because the accumulation process has a particular property. It begins before you are consciously aware of it. The neurons start climbing, the bucket starts filling, and you, the person who will eventually experience the choice as yours, are not yet aware that anything has happened. The question of when you enter the picture and what role you actually play turns out to be one of the most unsettling discoveries in this field. It was made not by studying value or selection or accumulation, but by asking the simplest possible question: at what exact moment does a person decide to move their hand? Benjamin Libet was a neuroscientist at the University of California, San Francisco, with a long-running interest in the gap between brain events and the experience of those events. He had spent years studying what happens in the brain during conscious sensation, and by the early 1980s he'd come to believe there was something worth measuring in the simplest possible voluntary act, the decision to move a hand. The readiness potential had already been described 2 decades earlier by a pair of German physiologists named Hans Helmut Kornhuber and Luda Dieker. They had recorded from the scalps of volunteers and found a slow, steady build-up of electrical activity beginning roughly a second before a voluntary movement. They called it the Bereitschaftspotential, the readiness potential. The brain was clearly preparing before the action happened. What nobody had asked was whether the person was aware of this preparation while it was building, Libet asked. His method was straightforward but demanded precision. Subjects sat in front of a screen showing a dot moving around a clock face, completing a full revolution in just under 3 seconds. The subjects were instructed to flex their wrist at whatever moment they spontaneously felt the urge to do so. There were no cues, no instructions about timing. The movement was to be freely chosen. And at the moment they first became aware of the intention to move, they were to note where the dot was on the clock face. That clock position gave Libet a timestamp, the moment when the subject first consciously experienced the urge to move. He compared it to 2 other timestamps. The first was the EEG record from the scalp, showing when the readiness potential had begun. The second was the EMG record from the wrist, showing when the muscle actually moved. The readiness potential began, on average, about 550 milliseconds before the movement. The subjects reported first becoming aware of the intention to move about 200 milliseconds before the movement. A gap of roughly 350 milliseconds separated the start of the brain's physical preparation from the first moment of conscious awareness. Think of a long-distance runner settling into the starting blocks. The body is loaded, muscles compressed, weight forward, every tendon primed for explosive release. The runner is ready to go before the gun fires. The preparation and the launch are separate events. What Libet found was something structurally similar. The motor system had started building the movement nearly 600 milliseconds before the hand flexed. The person's awareness of intending to move came only at the 200 millisecond mark. By the time you experienced the intention, the preparation was already more than halfway done. Libet was careful with his interpretation. He noted that subjects could still stop themselves from completing the movement after the readiness potential had begun. They had been instructed to sometimes do exactly this, to begin the process and then abort it. He suggested that conscious awareness might still have a role, not in initiating the action, but in vetoing it before completion. What he called, with careful understatement, the possibility of a free veto. The experiment has been repeated and challenged many times in the decades since. Each replication has broadly confirmed the gap, but the basic finding has held. The brain begins preparing a voluntary movement before the person is consciously aware of intending to make it. The subjective experience of deciding arrives late. Something has already started moving in the dark. What this means for the question of who is actually in charge is not straightforward. And the answer Libet implied is not the only possible reading of the data. But the data do raise one question that cannot be avoided. If conscious awareness of intention arrives after the physical process has begun, what is the conscious experience of deciding actually for? The finding from Libet's laboratory has been cited, argued over, and reinterpreted more times than almost any result in neuroscience. And the most common response to it, that it proves free will is an illusion, is probably the least defensible reading of what the data actually show. What the data show is more specific than that, and more strange. They show that the subjective sense of deciding, the feeling of willing yourself to act at a particular moment, does not correspond to the beginning of the brain's physical preparation for that act. The physical preparation begins earlier. The conscious experience of choosing comes later. These two events are real, but they are not in the order the experience implies. What this does not show is that awareness is absent from the process. The 350 millisecond gap between the start of the readiness potential and the reported sense of intention is not evidence of a ghost making decisions behind a screen. It is evidence of the gap between when a physical process begins and when the brain's monitoring systems register it as a conscious event. Every sensation you experience is processed before it becomes experience. A light flash is registered by your retina and processed through your visual cortex before you're aware of seeing anything. The delay is normal. The delay is the system. Libet himself argued that conscious awareness, while arriving late to initiate the movement, might still function as a veto. In his experiments, subjects who were instructed to sometimes start the process and then abort it were able to do so. They could feel the intention forming and stop before the hand moved. He proposed that while you may not freely will your actions into being, you may still freely choose not to complete them. He called it, with deliberate modesty, a free will to veto, the power to stop what has already started. A decade after Libet published his results, a neuroscientist named Aaron Scherger offered a reinterpretation that opened another possibility entirely. Scherger had noticed that the readiness potential looks similar to the kind of gradual accumulation of noisy neural signals seen in the same decision neurons Shadlan had been studying. He proposed that in tasks where people are told to move whenever they feel like it, with no other cue or instruction, the movement may be triggered not by a deliberate internal decision, but by random fluctuations in motor preparation that occasionally cross threshold by chance. In this reading, the readiness potential is not a signal of a decision being made. It is the background noise of a system always partially prepared to act, occasionally tipping over the threshold without any deliberate intent behind the tipping. This interpretation remains contested, but it introduces something important: the idea that some of what we experience as spontaneous choice may be less like deliberate will, and more like the motor system occasionally tripping over its own readiness. The sense of having decided then arrives as a post hoc label for something that was already underway for entirely mechanical reasons. None of this settles the deeper question. What it does is relocate it. The question is no longer simply whether you initiate your actions before you are aware of intending them. The question is what kind of thing your awareness actually is in a system like this. Is it a passenger reading the navigator's notes after each course correction? Is it part of the correction mechanism itself? Is the distinction between those two descriptions real, or merely a product of the way we talk about minds? The brain's decision system runs on more than value signals and suppression gates. There is a component to every significant choice that Shadlan's dot motion experiments do not capture, a component that Libet's study of wrist flexions cannot reveal. It is something that shows up only when the decision system is damaged in a very specific way, and its absence produces one of the strangest states the human brain can fall into: the ability to reason perfectly about anything, and the inability to choose. Antonio Damasio was a neurologist at the University of Iowa when a patient came to him with a problem that Damasio could not find a name for at first. The patient, whom Damasio would later refer to in writing as Elliot, had undergone surgery to remove a tumour from his frontal lobes. The surgery had gone well. The tumour was gone. Elliot's language was intact. His memory was intact. His reasoning was intact. His IQ tested in the normal range. By every standard measure that clinicians had available, Elliot's brain was functioning as it should. Except that Elliot could not decide anything. Not the major choices in life, though those were affected too. The paralysis extended to the smallest things. Deciding where to eat lunch could consume an afternoon. Choosing which pen to use while filling out a form required extended deliberation. Elliot could generate endless reasons for and against any option. He could analyze the problem from multiple angles, weigh the considerations, articulate the trade-offs with clarity. He simply could not stop doing this and commit to one path. Damasio tested Elliot carefully. He laid out 2 possible meeting times and asked him to choose. Elliot produced a sustained analysis of the relative merits of each time slot. He factored in parking, appointments, daylight. Damasio finally interrupted and simply told him which time to come. Elliot nodded and agreed immediately. Without an external constraint, the deliberation had no flaw. It continued until something stopped it from outside. What had been removed along with the tumour, Damasio concluded, was the emotional input to the decision system. The region that was damaged in Elliot, the ventromedial prefrontal cortex, sits at the junction between the brain's reasoning systems and its emotional memory. It connects the prefrontal areas that handle working memory and deliberation to the limbic structures that generate and store emotional responses. When that connection was severed, Elliot retained the capacity to reason, but lost access to something the reasoning system had been relying on without his awareness. Damasio named what was missing the somatic marker. A somatic marker is a bodily state attached to the memory of a past outcome. A shift in heart rate, a tightening or easing in the chest, a change in skin conductance. When you've eaten at a restaurant that made you ill, the memory of that restaurant carries a marker, a faint physical unease that precedes any conscious recollection of why you disliked it. That physical response is information. It is the brain's shorthand for what similar situations have previously led to. Think of a vendor who has been working the same market for 30 years. When a customer names a price, the vendor does not run through the economics from first principles. Something in the body already responds, an ease or a resistance before any formal calculation has formed. That response is not sentiment clouding the judgment. It is 3 decades of compressed experience, encoded not in words or numbers, but in the physical state the situation produces. Damasio's argument was that human decision-making works the same way. The somatic marker arrives before reasoning, and it biases the accumulation in the basal ganglia before deliberation can begin. Without this input, deliberation becomes symmetrical. Every option is as computationally valid as every other. The accumulation process has nothing to start from. The system cannot find a place to begin tipping. Eliot's condition was not just a curiosity of clinical neurology. It was a demonstration that the received picture of rational decision-making, the picture in which clear thinking produces good choices and emotion, is the enemy of reason, is precisely inverted. Emotion is not noise in the decision system. Emotion is one of the primary inputs to it. Remove the emotional signal entirely, and rational deliberation does not improve. It becomes incapable of terminating. What this means is that the prefrontal cortex, which sits above all of this circuitry, is not simply a reasoning engine. Its role in the decision system is more specific and more peculiar than that picture allows. There is a structure sitting directly behind your forehead, roughly the width of your palm, that does something no other region quite manages. It does not move your muscles. It does not store your memories in any lasting way. It does not feel your emotions or generate your desires. What it does is hold a representation of a situation in mind long enough for the rest of the brain to act on it coherently. The prefrontal cortex is the most recently expanded region in the human brain. It takes longer to mature than any other structure, still developing into your mid-20s, and the puzzle it solves is one that only becomes visible when it fails. Patricia Goldman-Rakich spent decades at Yale mapping what the prefrontal cortex actually does. In the 1980s and 1990s, she recorded from individual neurons in monkeys while they performed a simple task. The monkey saw a dot appear briefly on a screen, then the screen went blank. After a delay of several seconds, the monkey had to look toward where the dot had been. No cue remained. The location had to be held somewhere inside the brain itself. Her recording showed something unexpected. Neurons in the prefrontal cortex fired continuously during the delay, long after the dot had vanished. They were not responding to anything in the outside world. They were maintaining an active representation of something that was no longer there. Goldman-Rakich called it working memory, the capacity to hold information in mind and work with it, even in the absence of the stimulus that prompted it. Think of a conductor standing in front of an orchestra. The conductor does not play an instrument. During a performance, the musicians around them are producing all the sound. But the conductor holds the full structure of the piece in mind while it unfolds. They know what the strings just played and what the brass must do next. They know how far into the movement the orchestra is, how the tempo has been running, whether the balance between sections is right. Without the conductor's held representation, the musicians would still produce notes, but those notes would not add up to a performance. The prefrontal cortex works like that. It holds the structure of the current situation, while the motor systems, the emotional circuits, and the memory systems do the generating. What is relevant here? What the goal is? What happened recently that bears on this moment? The prefrontal cortex does not make decisions in isolation. It provides the context that shapes what the competing proposals in the basal ganglia are competing for. Damage to this region produces a specific and striking pattern. Patients retain general intelligence. They can solve logic puzzles and recall past events. They name objects and speak fluently. But they struggle to maintain a goal across time. They're easily captured by whatever is immediately present. They begin a task and follow a distraction instead, not because they forgot the task, but because the representation of the goal lost its grip before the task was finished. This is not memory failure in the ordinary sense. It is a failure to sustain context. The information is available. The system that holds it active long enough for the rest of the brain to orient toward it is impaired. What this means for decision-making is that the prefrontal cortex is doing something upstream of the moment of selection. Before the basal ganglia can select one action proposal over the others, those proposals need to be weighted not just by past reward history but by what matters right now. A decision made without prefrontal context is one shaped only by habit and immediate desire. A decision made with it is one that can take account of what you're trying to do, what you've already done, and what consequences you have reason to anticipate. The prefrontal cortex, in other words, is where longer time horizons enter the computation. It is the part of the brain that allows a choice made today to be shaped by a goal that will not be reached until tomorrow, or next year, or decades from now. Not because it stores the future, but because it holds the current situation in contact with what has been learned about outcomes across time, and underneath that capacity something stranger is operating, something the brain does not fully account for to itself. Michael Gazzaniga was not expecting the brain to lie to him. In the 1960s, he worked alongside Roger Sperry at Caltech on patients who had undergone a radical surgery for severe epilepsy. The corpus callosum, the thick cable of more than 200 million fibers connecting the two cerebral hemispheres, had been cut. The surgery stopped the seizures, but it also produced something strange. Gazzaniga developed a method for showing different information to each hemisphere independently. Because the left visual field feeds the right hemisphere, and the right visual field feeds the left, he could flash images or words to one side while keeping the other in the dark. And because the surgery had severed the line of communication between hemispheres, what one side received, the other genuinely could not know. When he showed a picture to the right hemisphere and asked the patient to pick up an object matching it, the patient reached with their left hand and chose correctly. When he then asked the patient why they had picked up that object, the verbal reply came from the left hemisphere, which had seen nothing, and the left hemisphere did not say it did not know. It invented an explanation on the spot, confident, fluent, and completely wrong. Gazzaniga called the mechanism the interpreter. The left hemisphere, which controls language in most people, does not simply report what happened. It constructs a story about what happened. It takes actions already completed, sensations already registered, then it weaves all of this into a narrative of reasons. The sense of having decided is not the decision itself. It is the story generated about the decision afterward. Gazzaniga pushed the experiments further. In one trial, he showed the right hemisphere a picture of a snow scene and the left hemisphere a picture of a chicken's claw. He then laid out a set of cards face up and asked the patient to point to the most relevant image. The left hand, controlled by the right hemisphere, pointed to a shovel. The right hand, controlled by the left hemisphere, pointed to a chicken. Both choices were correct for what each hemisphere had seen. Then Gazzaniga asked the patient why they had chosen those objects. The left hemisphere, which had seen only the chicken claw and knew nothing about the snow scene, did not say it did not know why the left hand pointed to a shovel. It said, "I chose the chicken because of the claw, and you need a shovel to clean out the chicken shed." The explanation was coherent. It was fluent. It connected the two actions into a single plausible narrative, and it was entirely fabricated, constructed in the moment by a hemisphere that had no access to the information that had actually driven one of the two choices. The left hemisphere was not lying in any ordinary sense. It was doing what the interpreter always does, completing an incomplete picture with the most reasonable story available. This does not mean the left hemisphere is deceiving you deliberately. It is doing what it has always done, making sense of the world by building coherent accounts of events. The problem is that the events it is accounting for were sometimes initiated by systems it cannot see. When those systems act, the interpreter narrates. The narrator assumes it was the author. Now consider what this implies for the moment before you made a choice. The accumulation process in the parietal cortex runs to threshold. The basal ganglia selects and releases the winning proposal, the motor cortex fires, the readiness potential rises, the body moves, and somewhere in the half-second before the movement, or perhaps just after, a sensation arrives. The sense of having chosen, the sense of being the one who decided. That sensation is real. It is a genuine experience. But it is arriving as a report about a process that was already largely complete. The feeling of authorship is accurate in the sense that the whole system is yours. All the machinery that ran the accumulation, performed the selection, and weighted the options by value belongs to you. It was shaped by your history and carries your particular patterns of reward and loss and inference. But the felt moment of deciding is not the cause. It is the recognition. What Gazzaniga showed is not that you do not make choices, it is that the machinery of making them runs below the level of the story you tell about them. Consciousness, in this picture, is not absent from decision-making. It enters the process genuinely. The prefrontal cortex holds context consciously. Emotion registers consciously. The veto Libet described operates consciously. But the felt sense of authorship is itself constructed after the fact, built by the same hemisphere that will tell you why you chose the way you did. There is something unsettling about this, and something clarifying. Go back to the moment the episode opened on. Your body had settled into whatever it was resting against. Your breathing had already slowed. At some point the signal to keep listening was sent and you kept listening. None of those registered as decisions. They felt like the body knowing what to do without being asked. Now you have the machinery underneath those moments, and what it shows is not what the common picture of decision-making suggests. The common picture places a conscious, deliberating self at the center of the process. You weigh the options, you evaluate, you select, you act. Consciousness comes first, action follows. The felt experience of deciding is the mechanism. The brain's record, assembled over the last century of neuroscience, looks different. Multiple systems run simultaneously, competing and cooperating in parallel, each contributing something to the outcome before the conscious experience of deliberation has begun to settle on anything. The timelines do not match the story. The felt moment of deciding arrives after the mechanisms that produce the decision have already done their main work. What the neuroscience shows is a different arrangement. Competing action proposals are already being constructed before deliberation begins, driven by sensory input and prior context. The basal ganglia is running a continuous selection operation, suppressing competing proposals and releasing a winner through a gate that opened because the direct pathway overpowered the indirect one. Dopamine has already been assigning predicted values to those proposals, values shaped by every similar situation your brain has ever resolved. The accumulation process has been ramping toward a threshold that, once crossed, commits the system to an action. The readiness potential has been building. The body has begun preparing, and then awareness arrives, entering a process that is already well underway. The question this raises is not whether you made the choice. You did. The question is what making it consisted of. And the answer, given everything the researchers uncovered, is that making a choice is not a single moment of conscious evaluation. It is a process distributed across hundreds of milliseconds and a dozen interacting systems, all of which are yours. Your history of reward and loss is encoded in those dopamine weights. Your past experiences are compressed into the somatic markers that made certain options feel right before you had articulated why. Your goals and your sense of the current situation are held by the prefrontal cortex as an active representation shaping what the entire system is selecting for. The patterns that ran all the way from affordance competition to threshold crossing are not external forces acting on you. They are you, operating at a depth that conscious experience cannot fully see. Consciousness does not stand apart from this and watch it happen. It holds context, it vetoes, it integrates, it narrates. But the narration it produces is generated after the fact, by a system making the best causal story it can from the evidence available to it. That confident sense of having weighed and chosen and decided is real. It is simply not the mechanism. And that story is mostly right, in the way that a good account of something complex is mostly right. Accurate about the conclusion, less certain about the exact sequence that produced it. There is something clarifying about this picture. Once the initial strangeness settles, you're not less the author of your choices because the mechanisms that produce them are partly invisible to you. You're no less the author of a sentence because you cannot see the path from intention to grammar to phonation. The process goes deeper than the experience of it. That depth is not a threat to the experience. It is the substrate the experience arises from. What the brain is doing in the moment you feel yourself deciding is recognizing a conclusion it has already reached. The gap between the process and the experience of it is not a flaw in the design. It is what the design produces. Consciousness is the brain's way of reporting on itself, and like any reporting system, it captures some things clearly and omits others. What it omits mostly is the machinery. It is the system becoming aware of what it has done. And that recognition, that felt sense of authorship, is itself a real event in the nervous system. It is not an illusion. It is the brain, which is you, discovering its own outputs. You made a choice tonight choosing to listen to this. The sense that you decided to do it arrived slightly after the process that made you do it. Both are true. Neither cancels the other. And the organ doing the recognizing is the same organ that ran the computation. It is the same organ that is now gently beginning to slow